For the objectives, the study aims to know if a host factor human TRA2A will able to bind to the ISS motif of either the mRNA of avian IAV or 3NS mRNA of the human IAV and will inhibit their splicing. To know if TRA2A mediated alternative mRNA splicing is crucial for virus pathogenicity and adaptation of influenza virus to humans.
So for the introduction, first is himay-himayin muna natin yung mga variables na included sa title nung study na napili natin. So the title is Human TRA2A determines influenza A virus host adaptation by regulating viral mRNA splicing. So when we say TRA2A, this is also called as the transformer 2 protein homolog alpha, which is a human protein that is encoded by TRA2A gene. So this is also a member of the transformer 2 homolog family and encodes a protein with two RS domains and an RRM or the RNA recognition motif domain. So this phosphorylated nuclear protein binds to specific RNA sequences, thereby plays a role in the regulation of pre-mRNA splicing. So for the influenza A virus, it can be classified into subtypes based on the presence of the two major surface glycoproteins, which is the hemagglutinin and neuraminidase. So for the H antigen, this is used to bind to host cells. So there are 16 H antigens from H1 through H16, and the human infections that usually occur with this H antigen is with H1, H2, and H3. While for the N antigen, this cleaves budding viruses from infected cells. So there are 9 N antigens from N1 through N9, and the human infections usually occur with N1 and N2. So under influenza A virus, there are several strains associated with it. Pero dahil nga sa study na to, it gives focus on avian influenza. So dun lang din tayo magfo-focus. So kapag sinabing avian influenza, it refers to the disease caused by the infection with avian influenza type A viruses. So when you say avian, it means bird. So mainly yung spread ng disease na to or ng virus na to is between birds lang. And these viruses occur naturally among wild aquatic birds worldwide. Pero tulad ng nung sabi ko kanina, since influenza viruses are zoonotic in nature, so it's very possible na the virus from the birds can be transmitted to humans also. So it's very frequent that IAVs from their nature reservoir may spill over to mammalian species. Pero the problem here is limited lang sa mga IAVs na yon are able to adapt or nakakasustain sa bago nilang environment. So, the key to the persistence of the influenza virus is its antigenic variation because the subtypes H and N antigens of influenza viruses is continuously changing. So, paano ko nasabi? Kasi merong dalawang types yan. The antigenic drift and the antigenic shift. So, the antigenic drift, it happens annually. So, it is caused by RNA replication errors of the virus. So, these RNA replication errors, it causes lang minor change in the antigenic structure of the virus. And it occur with all three types of influenza, the A, B, and C. While for the antigenic shift, this happens if merong change drastically in the surface antigen of the virus, causing major change. And if there is a major change, it results to the formation of the new H or N antigen. And this is only associated with influenza A. There are two mechanisms under antigenic shift. These are the reassortment and the adaptive mutation. So this reassortment occurs when the genome of different influenza viruses becomes mixed into a single virion, which results in the formation of a new strain of influenza virus. While for the adaptive mutation, this new strain or this novel virus, it can slowly adjust, and when it adjusts, it can be transmitted to mammalian host including human. And it is expected na since it's a new strain nga, it's a novel virus, the human population is likely to have little or no historic exposure or resistance to the new strain. And as a result, it increases the risk of pandemics. So, in addition lang, elaborate ko lang tong mga pandemics na nag-occur during the 20th century that is associated with the major shifts of influenza A virus. Pero binanggit din naman siya sa study, pero although year lang, i-elaborate ko lang ng onti kung ano yung mga viruses na yon. So, in 1918 to 1919, the virus H1N1, so it is designated as an influenza A virus subtype that has an H1 protein and an N1 protein. That is why H1N1. This is also called the Spanish flu, and it killed approximately 20 to 50 million people. And in 1957 to 1958, the virus H2N2 emerged, or the Asian flu. In 1968, the virus H3N2, or the Hong Kong flu, followed by the year 1998, which is the virus H5N1. So, ito na yung avian flu. This is in this, ito yung andito sa study. H5N1 siya. So, tulad ng sabi ko kanina, it acquired after direct or close contact with infected birds, and it is considered as highly pathogenic avian virus. And yung pinaka-latest lang is nung 2009. This is a highly infectious novel form of influenza caused by influenza A virus H1N1 emerge. So, this is the swine flu. So, 'di ba, ang daming pandemics na nangyari na associated sa influenza A virus, pero still the detailed underlying mechanism for these adaptive changes in IAVs are still not completely understood. But in this study, the researchers revealed that a host factor, which is yung nabanggit ko kanina, the human TRA2A, depresses mRNA splicing by binding to the ISS motif. So, ISS stands for the intronic splicing silencer. So, it's a splicing regulatory element, which is an important part in mRNA splicing. Since once it binds to the cis-acting RNA, it either positively or negatively influences the splicing. So, going back, so once na mag-bind na yung human TRA2A to the ISS motif of either the mRNA of avian IAV or the nsMRNA of human IAV, it inhibits their splicing, but with opposite effects on virus replication of human and avian IAVs in vitro and in vivo. So these results indicate that TRA2A mediated alternative mRNA splicing is crucial for virus pathogenicity and adaptation of influenza virus to humans.
For the third part, which is the methodology, starting with the materials, which will include the cells and viruses used in the study, as well as the antibodies and reagents used. Next step is the co-immunoprecipitation and Western blotting. Next is RNA in vitro transcription, labeling, and pull-down assay. Then RNA purification, followed by RNA EMSA. Then the fifth step will be indirect immunofluorescence assay and confocal microscopy. Next will be growth kinetics of viruses. And the eighth step is the fluorescence in situ hybridization. Then pathogenicity study in mice, and lastly the polymerase assay. For the cells used in this study, first one is the HEK, or human embryonic kidney 293T, which are cells that are taken from an aborted female fetus and is used because it can efficiently replicate influenza virus. Next, A549 are cells that came from human Caucasian lung carcinoma that are infected either by avian YS or H5N1, most commonly known as the bird flu, or by human PR8 or H1N1, the human influenza. The third cell is U251, a human glioblastoma cell line harvested from the human brain, which can also replicate influenza virus. Next is DF1, a continuous cell line from an embryo fibroblast, and DEF or duct embryonic fibroblasts. Both DF1 and DEF are infected by avian YS or H5N1. And lastly, MDCK or maiden Darby canine kidney cells are mostly used because of their high susceptibility to infection to various influenza virus strains. All cells were purchased from American Type Culture Collection located in the United States of America. Then, for the mediums used in the study, we have RPMI or Roswell Park Memorial Institute 1640 medium, second Tulbecos Modified Eagle medium, and lastly HUMS or F12 medium. These mediums are supplemented with 10% heat inactivated fetal bovine serum and incubated in 37 degrees Celsius humidified incubator with 5% carbon dioxide. Again, for the host factor, the study used human TRA2A or HuTRA2A. And for the viruses used in the study, they used human PR8 or H1N1 or the human influenza, and the avian YS or H5N1, most commonly known as the bird flu. 8 segments of a doc, Shenyang 1, 2005, H5N1 YS were cloned into PHW 2000. The letter A stands for influenza virus A, and the doc is the host of the virus, and Shenyang is the location where the virus were obtained. Another 8 segments of A Puerto Rico A34 H1N1 PR8 were cloned into PHW 2000. Again, H1N1 is the human influenza, and Puerto Rico is where the location where the virus were obtained. And lastly, mutant M gene at position 334 and NS gene at positions 234 and 236 were generated using a PCR-based site-directed mutagenesis.
Plasmids and small interfering RNA oligonucleotides. For this study, the plasmids that were used are flag SF2 and flag HNRNPK. The full-length cDNA of HuTRA2A and influenza viral PA, PB1, PB2, and NP genes of YS, H5N1, and PR8, H1N1 were amplified by the use of RT-PCR, and then they were cloned into P3X flag CMV, HAPCAGGS, and pcDNA 3.1 CMV respectively. For the A549 and U251 cells, they were transfected with each small interfering RNA using lipofectamine 2000 in accordance with the user manual for 24 hours before subsequent experiments. Antibodies and reagents. The antibodies that were used in this study are the following: rabbit anti-TRA2A, mouse anti-flag, mouse anti-HATAG, mouse anti-glyceraldehyde 3-phosphate dehydrogenase, rabbit anti-AAVPA, PB1, PB2, NP, NS1, M1, NEP, M2, and HA. Mouse anti-NP. Control rabbit IgG polyclonal. Horseradish peroxidase conjugated anti-mouse and anti-rabbit. Alexaflor 594 conjugated goat anti-rabbit. FITC goat anti-mouse, and FITC goat anti-rabbit. For the reagent, 4,6-diamidino 2-phenylindole with a ratio of 1:1,000 was used.
For co-immunoprecipitation and Western blotting, before we head to the step-by-step procedure, we should be able to understand first why are they being used as methods for certain studies. With regards to co-immunoprecipitation, it is utilized to identify protein complexes and determine protein-protein interactions, whereas with Western blotting, this technique can be a supplement as it identifies specific proteins from a mixture of proteins that were initially extracted from cells. And with this, for us to be able to grasp the particular principles for clinical applications, we have this journal article which utilizes these said methods. For co-immunoprecipitation, we have here the three processes. First is the reagent preparation, second is the immunoprecipitation, and third is the Western blotting for analysis. So in reagent preparation, we have here the HEK293 T cells, which is used for the production of retroviral vectors, and then the PR8 or YS virus, which is a particular strain of influenza A virus, and cold phosphate buffered xylene to maintain constant pH to prevent the cells from rupturing and shriveling due to osmosis. And then we also have here the cell lysis buffer, which breaks the cell membrane by changing the pH. And then the protein antibody bead from the word itself, and antibody for protein, and then immunoprecipitation buffer, and lastly ribonuclease A, which is used to hydrolyze RNA from protein samples. Moving on to the co-immunoprecipitation process, we have here three steps. So first is to harvest and wash cells, second is to prepare cell lysates, and then lastly is the immunoprecipitation. For the process of harvest and wash, we need first to infect the HEK293 T cells with free nucleic acids from both the influenza PR8 and YS virus strain for 48 hours. And then transfer the cultured cells from the culture dish to a 15 ml conical tube and centrifuge at 500 times gravity for 2 minutes at 4 degrees Celsius. Wash with cold phosphate buffered xylene or PBS and centrifuge at 500 times gravity for 2 minutes at 4 degrees Celsius to remove the supernatant. For cell lysates preparation, this is an important process because the yielded proteins will be used for analysis. So in this process, we suspend the cell pellet in cold cell lysis buffer containing protease inhibitor cocktail to prevent protein degradation after cell lysis, and then incubate it on ice for 10 minutes and centrifuge at 13,000 times gravity at 4 degrees Celsius for 10 minutes, and the supernatant yielded will be transferred to a fresh tube. For the last process of co-immunoprecipitation, on the fresh tube with cell lysate, add the antibody against the TRA2A and incubate at 4 degrees Celsius overnight. Transfer the immunocomplex obtained to a new tube which consists of lysate and antibody solution. After that, add the antibody bead to the immunocomplex and let them precipitate for 4 degrees Celsius for 1 hour. Wash the protein antibody bead complexes 3 times with immunoprecipitation buffer before boiled in SDS loading buffer and analyzed by Western blot. Treat the complexes with ribonuclease A at 37 degrees Celsius for 1 hour to remove unspecifically bound RNAs. So moving on, for us to be able to identify the specific proteins from the protein complexes, we have here the Western blotting, which has several processes, which includes the sample preparation, the gel preparation, electrophoresis, electrotransfer, and blocking and antibody incubation. So for the sample preparation, we have to determine first the volume of protein extract to ensure 50 micrograms in each well, and then add 5 microliter sample buffer to the sample. And then heat the samples with dry plate for 5 minutes at 100 degrees Celsius, which speeds up the process of denaturation by increasing molecular motion. So for gel preparation, first is we have to assemble the rack for gel solidification. Next, add stacking gel solution until the level is equal to the green bar holding the glass plates. Add water to the top to remove impurities and wait for 15 to 30 minutes until the gel solidifies. Overlay the stacking gel with the separating gel after removing the water. So the stacking gel and the separating gel are two types of polyacrylamide gels which is used to get better separation of protein molecules in a given sample. So insert the comb which is used to create the wells in gels for electrophoresis. Make sure that there are no air bubbles and wait until the gel is solidified. Moving on to the process of electrophoresis, first is pour the running buffer into the electrophorator and then place gel inside the electrophorator and connect to a power supply. Make sure that the buffer covers the gel completely and remove the comb carefully. Load markers 6 microliters followed by samples 15 microliters into each well and then run the gel with low voltage 60 voltage for separating gel and use higher voltage for stacking gel which is 140 voltage. Lastly, run the gel for approximately an hour or until the dye front runs off the bottom of the gel. So for electrotransfer, first is we have to cut 6 filter sheets to fit the measurement of the gel and 1 polyvinylidene fluoride or the PDVF membrane with the same dimensions. So the 6 filter sheets are composed of 2 fiber pads, which is also called the sponge, and 2 filter papers, the membrane, and the gel. Second step is we need to wet the sponge and filter paper in transfer buffer and wet the PDVF membrane in methanol. Separate glass plates and retrieve the gel and create a transfer sandwich. So after we have created now the sandwich, we have to relocate it to transfer apparatus which should be placed on ice to maintain 4 degrees Celsius. Add transfer buffer to the apparatus and make sure that the sandwich is covered with the buffer. Place electrodes on top of the sandwich and ensure that the PVDF membrane is between the gel and a positive electrode and transfer for 90 minutes. So for the last process of Western blotting, we have here the blocking and antibody incubation. So blocking is a very important step of Western blotting as it prevents antibodies from binding to the membrane non-specifically. So for the first step, we have to block the membrane with 5% skim milk for 1 hour. Add primary antibody in 5% bovine serum albumin or BSA and incubate overnight in 4 degrees Celsius on a shaker. The BSA blocking buffer is ideal for saturating excess protein binding sites on membranes and microplates. Wash the membrane with TBST for 5 minutes. So the Tris buffered saline 0.1% twin 20 detergent is a wash buffer. And then add secondary antibody in 5% skim milk in TBST and incubate for 1 hour. Wash the membrane with TBST for 5 minutes and prepare enhanced chemiluminescence mix and incubate again the membrane for 1 to 2 minutes and visualize the result in the dark room. So again, for us to better understand the Western blotting analysis, we have here a simplified protocol of the technique. First is prior to loading, add buffer to samples and then load the samples onto a gel, and then proteins are electrophoretically transferred to a membrane support. Remove the membrane and incubate the blot with antibody. Detect and visualize the protein. Moving on to the next procedure, we have the RNA in vitro transcription. labeling and pull-down assay. So for the reagent preparation, first is we need to have segments for cloning. So we have here the full-length M and NS segments. And then we need to have the vector, which is the pcDNA3, the restriction enzyme XBA1, which is used to cleave foreign DNA, thus eliminating infecting organisms. The T7 RNA polymerase is an active enzyme that synthesizes RNA, and the DNase, which is used to degrade DNA, Pierce RNA 3' endes thiobiotinylation kit, A549 cell nuclear extracts, and Pierce magnetic RNA protein pull-down kit. So for the process of RNA in vitro transcription, we have to clone either full-length M segment, which is composed of 1-333, 1-360, and 1-360 mutant, or full-length NS segment, which is also consists of 1-232, 1-256, and 1-256 mutant into pcDNA3 that contains the T7 promoter. Linearize cloned plasmids with restriction enzyme XB1A and add T7 polymerase to synthesize mRNA by in vitro transcription. Add DNase to degrade DNase and transcribe mRNA into cDNA by reverse transcriptase enzyme. Label mRNAs with biotin using Pierce RNA 3' endes thiobiotinylation kit and incubate labeled probes and biotin and labeled RNAs with A549 cell nuclear extracts. So in the process of RNA pull-down assay, we need to wash 150 microliters of streptavidin agarose beads with buffers. So streptavidin agarose is used for fast and specific binding and separation of biotinylated molecules. Next is pre-clear nuclear extract by incubating with 60 microliters of streptavidin beads for 30 minutes at 4 degrees Celsius with rotation. Spin at 2,500 revolution per minute for 30 seconds and add 20 micrograms of the biotinylated RNA to the pre-cleared nuclear extract and incubate at 4 degrees Celsius for 1 hour in a rotary shaker. And for us to isolate biotinylated RNA bound to protein, add 30 microliters of washed streptavidin agarose beads per condition and further incubate at 4 degrees Celsius for 45 minutes. Wash beads 5 times with buffer. So again, for us to understand what is really the RNA pull-down assay is, here is a simplified protocol of the technique. So first, we need to label the RNA with biotin and then capture the labeled RNA magnetic beads. So from the previous slides, we use streptavidin beads. And then we need to wash the unbound materials and then elute and then detect. So after performing the RNA in vitro transcription and RNA pull-down assay, the proteins will be then analyzed by Western blotting, which was previously discussed.
For the process of RNA electrophoretic mobility shift assay or RNA EMSA, after 24 hours, the HEK293T cells were harvested and washed by phosphate buffered saline or PBS and lysed with sucrose buffer containing NP40. The nuclear fraction of the HEK293T cell was spun down and washed by sucrose buffer without NP40. The nucleus pellets were resuspended in the low salt buffer and incubated with an equal volume of high salt buffer for 30 minutes on ice. The NEs was prepared by spinning at 16,000 for 15 minutes, and for the binding reaction, 10 nanometer of RNA probes labeled with CY5 at 5 end were incubated with increasing doses of NE in 20 microliter of buffered mixture. After incubation for 30 minutes at room temperature in the dark, mixtures were separated by 6.25% non-denaturing gel, which consists of 0.5 trisporate EDTA. The gels were scanned by an fluorescent image analyzer.
For RNA purification, the journal used triazole reagent to isolate the total RNA from A549 cells, eliminating the DNA through the use of DNase treatment. Then, the RNA was transcribed to cDNA or circular DNA by reverse transcriptase. RT-qPCR using SYBR green 1 and semi-qPCR using primers targeting M1 and M2 gene was performed. Then, GAPDH expression was identified for each sample for the normalization of gene expression.
Moving on to fluorescence in situ hybridization. First, to detect PR8H1N1 M segment vRNA, fish probes with quasar 570 fluorophore were used. A549 cells infected with PR8 or YS virus at an MOI of 5 were put in a 12-well plate. It was then put to fix with 4% paraformaldehyde for about 10 minutes. Washing was done with PBS for a total of 3 times. Afterwards, 0.2% Triton X100 was used to permeabilize the cells for 10 minutes and were washed briefly. It was then incubated for 5 minutes with 200 microliters of wash buffer A. Next, wash buffer A was removed and 300 microliters of hybridization buffer with 3 microliters of fish probes was added. This was then incubated at 37 degrees Celsius in the dark without sunlight for 16 hours. This time, the hybridization buffer was again removed, and then using wash buffer A, incubation took place for 5 minutes at 37 degrees Celsius. Lastly, a confocal microscope was used to obtain images. For the pathogenicity study in mice, groups of 5-6-week-old female mice were lightly anesthetized with carbon dioxide. Next, inoculation was done with tenfold 50 microliter serial dilutions. Daily monitoring was done for weight loss and mortality for a total of 14 days. MLD50 values were calculated using the Reed and Muenck method. Afterwards, groups of 3 mice were intranasally inoculated with the aforementioned doses diluted in PBS and euthanized on 3 and 5 DPI to determine virus replication. To determine virus titers in lungs collected from infected mice, chicken embryonated eggs were used. As for the polymerase assay, HEK293 T cells, grown in 12 well plates, were co-transfected with reporter plasmid Pol1 NS firefly, virus polymerase, and NP plasmids, plasmid PGL4.75, and flag NEPPR8 expressing plasmid, or an empty vector for 25 hours. Cell lysis occurred at 24 hours after the transfection. Next, firefly luciferase and vanilla luciferase activities were determined. Dual luciferase reporter assay system was used for this. Lastly, data were presented as relative firefly luciferase activities, normalized to vanilla luciferase activities, and were representative of 3 independent experiments. As for the statistical analysis, virus titers in mouse tissues were analyzed using one-tailed pair t-test. On the other hand, the virus titers in A549 cells were analyzed using two-tailed paired t-test.
Single mutation at M334 alters virus pathogenicity in mice. The researchers determined the pathogenicity of ISS mutated virus in bulb C mice, which are albino mice bred from the strain of the house mice. Results displayed that mice infected with YSM334 had enhanced pathogenicity, displayed severe weight loss and disease, and had 100% mortality. Also, mice infected with YSM334C or PR8WT had moderate to severe bronchoalveolar necrosis, pulmonary edema, and inflammatory cell infiltrates. The YSM334C virus titers were 20-fold higher than those detected in YSWT infected mice. However, on the other hand, those infected with PR8-MS334G had decreased virus pathogenicity. They had Rather, they showed slight weight loss and 50% mortality. Mice infected with PR8WT had severe diseases and a whopping 80% rather mortality. PR8WT virus titers were 30-fold higher than those detected in mice with PR8-MP34G. TRA2A prefers binding to the mRNA with ISS. Hence, TRA2A is correlated with virus replication and virulence. The destruction of ISS in M of the avian virus enhances viral pathogenicity in mammals, whilst restoring the ISS in M of the human virus reduces pathogenicity in mice. In addition, M334 is critical in adaptive mutation for avian influenza A viruses.
To investigate whether TRA2A affects viral replication at the early stage of infection, the researchers monitored the accumulation of virus proteins at 3, 6, and 9 HPI. Knockdown TRA2A was observed to increase the levels of PB1, NP, M1, M2, NS1, and NEP in YS-infected A549 cells. However, opposite results were observed by the researchers in PR8 infected cells. Then, the researchers observed increased ratios of YSM2M1 and PR8-NEP-NS1 proteins in C TRA2A cells when compared to those in C and C cells infected with their respective viruses. Although the researchers found that the protein ratios of YSNEP-NS1 and PR8M2M1 in both TRA2A and NSC knockdown cells remain constant. Abnormal ratios of YSM2M1 and PR8-NEP-NS1 proteins suggest that TRA2A might negatively regulate the YSM and PR8-NS mRNA splicing. Y2M2 M1 mRNA ratio was notably increased compared to YSNEP NS1 mRNA ratio, which was not altered in TRA2A knockdown cells, while on the other hand, PR8 M2 M1 mRNA ratio did not change, but the YSNEP NS1 mRNA ratio was not altered in TRA2A knockdown cells. The researchers then performed the same experiments on U251 cells and got similar results attained from the previous one, which was from A549 cells. The researchers also used a semi-quantitative method to detect splicing of mRNA, and the result showed that knockdown TRA2A was beneficial to YSM splicing but did not affect PR8M splicing. However, overexpression of CHTRA2A was not observed to change the YS viral proteins, indicating that Viral mRNA splicing was also not changed. The data gathered expresses that TRA2A specifically inhibits the YSM and PR8NS mRNA splicing, leading to a completely opposite effect between human PR8 and avian YS virus replication. The researchers determined how TRA2A targeted mRNA and inhibited splicing under virus infection. RIP and RT-qPCR was used by the researchers and showed that YSM mRNA was enriched more than eightfold by precipitation of TRA2A antibodies in infected A549 cells, in contrast to the control antibody IgG. While on the other hand, PR8NS mRNA was enriched more than fivefold in comparison with both U87 and PR8PB1 mRNA. The researchers also observed that the intron of both YSM nucleotides 334 to 343 and PR8NS nucleotides 233 to 242 carries an RRM that bears a purine-rich nucleotide motif GAAARGARR. Among the nucleotides, the position 334 in YSM is a guanine, whereas the according position in PR8M is a cytosine. There is a nucleotide difference in this motif between YSNS and PR8NS. The researchers made a series of truncations and mutations to determine whether TRA2A binds with the YSM and PR8NS mRNA through the motif GAAARGARR. The results indicated that the TRA2A binds the mRNAs of both PR8NS and PR8NS1 256, but not that of PR8NS1 232, PR8NS1 256, YSNS, and its truncations. Although TRA2A interacted with the mRNAs of both YSM and YSM1360, but not with that of YSM1333, YSM1360, and PR8M full and its truncations. These stated results showed that the motif in YSM and PR8NS is essential for their binding with TRA2A. To confirm that, the researchers performed an RNA pull-down assay. Then, the results indicated that TRA2A protein preferably bound to the PR8 and SWT, YSNS234A, YSMWT, and PR8M334G compared to the controlled pyrimidine-rich CU, cytosine and uracil, RNA. EMSA, or electrophoretic mobility shift assays, was also used to determine that the PR8 and SWT probes bound with more TRA2 proteins than YS and SWT. YSM334C mutation substantially decreased its binding to TRA2A in contrast to PR8M334G mutation, which showed the opposite results. The researchers also found out that HRNPK promoted the replication of the avian virus. RIP assay was also performed to determine if the binding of HNRNPK to mRNA is influenced by TRA2A. The results showed that TRA2A did not affect the binding of HNRNPK, but also demonstrated that the GAARGARR motif in YSM and PR8NS mRNAs is required for their association with HU-TRA2A, and mutation of this motif disrupts it.
Single mutation at M334 alters virus pathogenicity in mice. The researchers determined the pathogenicity of ISS mutated virus in bulb C mice which are albino mice bred from the strain of the house mice. Results displayed that mice infected with YSM334 had enhanced pathogenicity, displayed severe weight loss and disease, and had 100% mortality. Also, mice infected with YSM334C or PR8WT had moderate to severe bronchoalveolar necrosis, pulmonary edema, and inflammatory cell infiltrates. The YSM334C virus titers were 20-fold higher than those detected in YSWT infected mice. However, on the other hand, those infected with PR8-MS334G had decreased virus pathogenicity. They had they showed slight weight loss and 50% mortality. Mice infected with PR8WT had severe diseases and a whopping 80% mortality. PR8WT virus titers were 30-fold higher than those detected in mice with PR8-MP34G. TRA2A prefers binding to the mRNA with ISS. Hence, TRA2A is correlated with virus replication and virulence. The destruction of ISS in M of the avian virus enhances viral pathogenicity in mammals, whilst restoring the ISS in M of the human virus reduces pathogenicity in mice. In addition, M334 is critical in adaptive mutation for avian influenza A viruses.
So, determining the effect of the ISS motif in PR8NS on splicing, using reverse genetics to engineer ISS mutations in the background of PR8 virus. Mutations resulted in loss of the ISS and a notable reduction of the NP and NS1 mRNA and protein levels of NP and NS1. However, NEP and mRNA and also as well as protein levels were not changed, and that results to the enhancement of NEP or NS1 ratio, and thus results of growth kinetics showed that the 234G over the 236G substitutions in PR8 and has notably decreased virus replication as compared to the WT viruses in A549 cells. So that concludes that those results suggest that only a small amount of NEP is required for human uh, IAV replication, while excessive NEP inhibits viral polymerase activity. So those results together uh, demonstrates that PR8NS with the ISS increases 3A TRA2A binding, which is not that beneficial for its splicing, leading to less NAP protein production, thereby um, promoting human virus replication.
So transfer alpha to homolog affects viral replication because of regulating mRNA. The result that the study shows that mRNA splicing of both ISS mutated viruses did not change in the transfer alpha to homolog knockdown cells, and there's also no change in the transfer alpha to homolog knockdown cells, and as well as no difference in virus replication of the mutated YSM and PR8NS viruses was observed in the transfer alpha 2 homolog knockdown cells and the control cells. So the studies conclude that the transfer alpha to homolog affects viral replication because of regulating mRNA splicing. So that would be all. Thank you.